I'm going to talk a little bit about a new model that we're trying to push out of Northeastern University to encourage more data sharing for neglected tropical diseases. We've seen a lot today about how huge amounts of hit matter are now available through high-throughput screening campaigns being done in industry and academic labs, large numbers of compounds, and all this data, or a lot of the data, is publicly available through Chembl, for example, and a PubChem, Collaborative Drug Discovery public vault. And really what these are hit compounds, and we need to be clear about this, right? So these are compounds that meet some minimum criteria. There are some really high quality hits in there, but really they need to have this optimization process done. And I'm a hit-to-lead med chemist. I spent about 10 years in industry doing that. And I can tell you it's a very specialized skill set. And there are very few groups that are doing this around the world, particularly outside of the large research organizations at Dundee, for example, and within pharma companies. And so there are more and more efforts in this space, academics and nonprofits and CROs and such joining forces to do this kind of work. But the key questions here are, first of all, who's working on what? Because we're all looking at the same data. So who's doing what and who's working on what parasites? What's been done? What has not been done? What has failed? People don't publish stuff that fails, right? And so generally you don't see these so-called negative results. Importantly, are there gaps? Are there things that people miss? If I triage a high-throughput screening set and another chemist looks at it, Charlie, where are you, Charlie? You still here? Yeah, there you are, front row. If he and I look at the same data set, we're going to pick different compounds out of that set, right? Those are gaps. And the question is also, are the endpoints people are working on the same? If I'm working on sleeping sickness, am I using the same type of assay, for example? And how do I compare it with someone else's? And importantly, this feedback loop of these optimization processes, are these ending up back in the public domain or not? Do those end up sort of hidden behind firewalls? So when I was thinking about this, I said this box of folks working together is really what I would call a distributed drug discovery program, right? Everyone has their own role to play in these projects, and it's loosely coordinated in some cases or tightly coordinated in others. And we have some experience in distributed drug discovery because in my lab at Northeastern, we don't do anything but medicinal chemistry. All the screening is done in collaborator labs. We started it with the MBL with Bob Campbell. It's gotten bigger over the last 5 years. And now we have a large number of programs with pharma, with other academic labs, with government labs at Walter Reed, for example. Everyone's doing something different. And what's happening is we sort of could see this coming, and so we needed to have a way to share data in a really efficient way, and so we needed a flexible data system. And Excel is not a data system. What I needed was a chemist-proof database system. I don't know anything about Oracle. I don't want to talk about Oracle. I don't want nothing to do with it. You can do it. It needs to be low maintenance, and data needs to come in and go out easily, and it has to be relatively low cost. It has to be able to register compounds, import data, compute properties, and selectively share data with various collaborators. And so we settled on the collaborative drug discovery solution. It's cloud-based. We don't do anything except interface with it. We don't have to maintain it. This slide is courtesy of Collaborative Drug Discovery, and they're well established in the space. These are some of their customers around the country, and really, from an iterative process of medicinal chemistry, you need something that will register compounds. So every compound has its unique registration. You have a way to get biology and drug metabolism data in and out. You have to have a way to get the data out so you can then use third-party visualization tools, although this CDD now has more and more of these computational tools now integrated into the database. And so just to give you a snapshot of what this is, you probably all know what a database looks like, but chemical structures with biology data. The CDD Vault will also calculate your dose response curves if you like. So having all this stuff in a sortable way, an exportable way, was actually really important to us and has proven to be really useful. And importantly, you can control your projects on the basis of, you can control access to your projects on the basis of who needs to see what, who can read read the database, who can write the database. So it's very well-vetted technology for this purpose. And all of our collaborations go through the Vault. I don't collaborate with anyone who insists on Excel. So getting back to the original question, this picture here is a fantastic thing. But we don't have a lot of money to spend on this globally, right? We saw these numbers earlier today. It's kind of depressing. And so it's really a bad idea to continue working not knowing whether you're duplicating someone's effort, maybe not precisely, but closely, or missing opportunities to make the most of your data and your other people's data and find some kind of synergy. And so there are a few ways we can do it. One way we heard about just a couple moments ago, the fully open model, where it's completely open to anyone who wants to participate, is fully transparent, ideas are shared, everything is shared in real time. On the other side is the pharma model, the fully closed model. By and large, drug discovery is done within a company. And you can do this in a very high, when you're in an environment where you can do everything in your walls, this actually works really well. But if you have to collaborate with other people, it's a little bit tricky. And in the middle, what we're envisioning is a way in which we can share data with an expanded group of teams beyond our lab, beyond our immediate collaborators, share the most critical details, share resources and models, and figure out a way to advise each other without exposing undue risk, because there's a lot of perceived risk here. So open source drug discovery malaria is on the fully open side. I put pharma, because of industry, on the right. And in the middle is what we're calling a hybrid open, and we're still looking for a good acronym for this, but something catchy. So the way this works is this. So we have this knowledge store. This is a collaborative drug discovery vault. In there are structures and screening data and ADME data, as well as an ability to search against public data sets that are already out and available. And the important part about this having a confidential store is that people can put their data in there, and it's not a public disclosure. So one of the things that I realized when I left industry was you can talk about your data, like in public to people. At Pfizer, it was like, oh, no, you can't talk about that. And if you get pharma people like me into an academic environment, you get hives if people start asking you to ask for data, which doesn't make a whole lot of sense, right? So in this case, what we're looking for is a transition between the fully open at OSDD malaria and the fully closed model, trying to get people into the comfort zone of sharing data in a more open way. The way you do that is you put a firewall around it. You say, if you're putting your data in here, it's confidential. We're also going to have a file share, as well as shared, understood, targeted product profiles for each pathogens. This is where organizations like DNDi are super important in terms of giving us what are the goals we're shooting for, and as well as resources for key tasks. I'll get back to that in a second. And so the idea is that research teams from around the world could deposit their data, whether it be chemical structures, screening data, computational models, ADME, predicted properties, et cetera, deposit their data into one common database where anyone who contributes can see the data in the database. But you can't talk about it with other people, right? You can talk about it within the consortium, but you can't talk about it to the external environment. The idea is we would also have access to folks who are stakeholders. For example, people like MMV or Drugs for Neglected Disease Initiative could have access and see and help advise and help, because really the market for this, I think, are labs like mine, which are relatively small, 12 people, right? And there are groups like mine scattered all over the place who are working in this area that can really benefit from combining forces in this way and getting some real good feedback. We have a way, as I showed, of getting public data in, and in addition, one of the strengths of CDD, another one of the reasons we went with it, was once you publish data, it's very easy to release it to the public, into the CDD public vault. And so we're looking for participants, right? So if you go to my lab website, you can find the link and you can sign up with your details. And our first kickoff meeting, really for the whole initiative, is on Monday. So here's how it works. So the ground rules are as follows. All the data in the vault is confidential to the consortium members. Okay, I can talk about your data with you and the other people in the consortium, but I can't talk about your data in my grant proposals, in my papers, things like this. It's a confidential disclosure. We want people to disclose all their data, not just the good stuff and not just the stuff that didn't work, right? Both are really important. MedChem, without knowing what compounds are ineffective, you're wasting time and effort. So negative data, things that don't work, need to go in, and as close to real time as possible, not as it comes off the plate reader, but as the data has been, you have replicate experiments, you're comfortable with it, deposit the data with the structures. And there'll be no lurking. So no one can deposit a handful of compounds and then never do anything except scan everyone else's data, right? We're going to develop agreed upon metrics for how this behavior will be dealt with. We want people to come in and participate. We'll probably do some kind of a quarterly meeting where we share each other's projects, people give an update, try to keep a community growing in this area. And we will know who will the sneaky people be. We are not, and I've been accused of all these things, we are not trying to coordinate anything. We're not trying to consolidate efforts. We're not trying to, all we're trying to do is to get people to see what others are doing so you don't do the same thing. We're not forcing collaboration, and we are certainly not looking to rip anybody off. Direct quotes. The advantages are obviously, this is what I would call a safe venue to share your data and to actually say, look, these are the things we're missing. Has anyone seen this before? Has anyone worked on this chemotype? We're having trouble making analogs. Have you ever done anything like this? You can share the data. You can constructively share negative data. Geez, the SAR didn't make a whole lot of sense. It was really flat. Look at all these compounds we made. Does anyone have any thoughts? To be able to bring in public data and see it side by side with your data is really powerful, and to be able to publicly share it when you're ready. And then, in addition, most importantly, in my view, is an opportunity to identify really complementary efforts. For example, I'm working on some kinesins for T. brucei, and someone else is working on a similar kinase in T. cruzi. Why wouldn't we collaborate, right? Why would we not cross-screen our compounds? In fact, 2/3 of the projects in my labs came from that exact kind of approach. And so that cross-fertilization can be really, really powerful. So right now we're seeding the database. All the compounds that we have made and registered in our database at Northeastern in collaboration with all of those parasitology labs are going into the database. That's about 2,000 compounds, about 2/3 of which have been tested against Leishmania, have been tested against all 4 parasites that we work on: Leishmania, T. cruzi, T. brucei, and malaria. And this also includes a high-throughput screening set of hits, a set of high-throughput screening hits that we got out of our really nice collaboration with the Open Lab in Triscantos. We're going to publish most of those hits, but a lot of them are held back because Glaxo asked us to. And so those will be in this database for people to come and see expanded data and to actually contribute and hopefully deposit the new data that they generate with those compounds back in. As of today, we have 10 labs that have committed to a teleconference on Monday. Not all will join, I'm sure, but that's a really strong start. And we're looking to develop a mechanism to gap fund experiments. So if you're talking to folks who maybe have a very small group, they don't have any money to do an ADME experiment. They need to do a drug metabolism experiment. We're going to have funding available to get a gap filling experiment done, or a PK study, or a hERG assay, or something like that. The challenges are the idea of, okay, Pollastri, you first. That actually slows everything down, right? We want a, yeah, me too kind of feel. Funding is going to clearly be a challenge. We raised $25,000 by way of a crowdfunding campaign to get this off the ground, which covers the cost of the database for the participants. Plus, we have money left for the seed funding that I'm talking about. There are definitely different kinds of phenotypes of researchers, right? Some are really kind of, I don't know about this. And we have to sort of accommodate this various level of comfort. And really, as far as intellectual property is concerned, we believe that this is a solution to allow people to share their data. And because it's not a public disclosure, could potentially file a patent on it if they wished. This might actually make them feel better, right? It's essentially sharing data essentially under a mutual confidentiality agreement. So the sales pitch here is that if you're interested or you're curious about more, you can visit the site here and share your details. And I comment about this on Twitter quite a bit. I will leave it at this, except to thank the folks at Collaborative Drug Discovery, CDD, Barry Bunin, who's got a write-up in the booklet, Sean Eakins as well, who's really helped me develop this model and come up with some ideas. We have 56 donors that donated $25,000. To a campaign at the last quarter of last year, which we're very grateful for, and Northeastern University for funding a lot of this work. So thanks again for inviting me.